pluripotent stem cells derived from dead embryos. Under this proposal, viable embryos would be derived from early in vitro fertilization embryos of roughly 4 to 8 cells that have spontaneously died, as large percentages of embryos actually do. Crucial to this approach is defining and verifying what represents organismic death in an early embryo, which represents an ongoing challenge for ethics and science. This would be a determining factor in whether this approach could ever be used in humans, as Drs. Donald Landry and Howard Zucker of the Columbia University of Physicians and Surgeons presented this proposal to the Council. The preliminary evaluation from the Council in 2005 was that, while it raised some serious ethical questions, the Council found the proposal to be ethically acceptable for basic investigation in humans as long as stringent guidelines like those proposed by Drs. Landry and Zucker were strictly observed. At the time, the proposal had not been tested even in animals. The Council suggested that certain non-invasive studies to explore and define organismic death could be pursued immediately. In June 2006, Dr. Landry published a peer-reviewed study that proposed applying the ethical framework currently used for obtaining essential organs from deceased persons for transplantation to the harvesting of stem cells from dead human embryos. The study presented a history of the concept of embryonic death and compared the function of viable embryos with non-viable embryos. Dr. Landry's research suggests that an irreversible loss of function could be documented and thus used as criteria that would permit a determination of embryonic death. Using this criteria, Landry made the case that approximately 1/5 of all embryos generated for in vitro fertilization could be reclassified as dead. This would enable thousands of new embryonic stem cell lines to be created without destroying a single living embryo. In September 2006, scientists from the Center for Stem Cell Biology and Developmental Genetics at the University of Newcastle in Britain published a peer-reviewed study that described how embryos lacking essential function could be used as a source for the derivation of human embryonic stem cells. Despite the promising work of Dr. Landry and the science at Newcastle, more work is required to address the concern that stem cells from dead embryos might not have the same pluripotent capacity as stem cells obtained from living embryos. In addition, more research is required on the concept and criteria that would permit determination of embryonic death in a way that is ethically unassailable for use in human embryos. B. Pluripotent stem cells via blastomere extraction from living embryos. Under this proposal, pluripotent stem cells would be obtained through biopsy of an early human embryo. Crucial to this approach is finding a stage of early embryonic development at which one or few cells can be removed without harming the embryo, and with the removed cells being usable as a source of pluripotent stem cells. The preliminary evaluation from the Council in 2005 was that this proposal was ethically unacceptable for humans. The Council stated that imposing risks on living embryos destined to become children for the sake of acquiring stem cells for research could not be justified. While the approach could be attempted in animals, the Council did not anticipate that results from animal experimentation could alter its assessment that this method would be ethically unacceptable for humans. In August 2006, Dr. Robert Lanza and a team from the firm Advanced Cell Technology made widely reported claims that human embryonic stem cells could be derived from blastomeres without requiring the blastomere's destruction. However, once the details of the study's methodology was reported, it became clear that the techniques used by the researchers did in fact destroy every one of the human embryos used, a fact ACT did not make clear in its initial press releases. This work thus neither proved nor disproved that human embryonic stem cells could be derived from individual cells extracted from an embryo without harming it. C. Pluripotent cells derived from biological artifacts. Under this proposal, pluripotent stem cells lines could be derived from an engineered biological artifact that lacks the organismal character of a human embryo. Crucial to this approach is demonstrating both that the developing entity is truly not a human embryo and that the cells derived from it are normal human pluripotent cells. In addition, one must show that creating such biological artifacts does not itself introduce other ethical problems. One such proposal, altered nuclear transfer, or ANT, was presented at the Council by member Dr. William Hurlbert of Stanford University. The preliminary evaluation from the Council in 2005 was that this proposal would need to be carefully tested in animals before human trials could be considered, but that there were no insuperable ethical objections that would preclude pursuing it. The possibility of any future endorsement for trying this approach in humans would depend upon a more thorough ethical analysis made possible in part by the animal experiments. In October 2005, Rudolf Janich and Alex Messner scientists at the Whitehead Institute for Biological Research at MIT conducted a peer-reviewed study of ant in mice. To assess the validity of ant, the researchers conducted a study that used a process of gene slicing and nuclear transfer to generate laboratory-constructed biological entity that could not implant in a uterus and was morphologically unlike a natural embryo. This non-embryonic entity nonetheless yielded fully functional pluripotent stem cells with the same characteristics as those obtained from embryos. The study in mice noted that this approach could produce pluripotent stem cells genetically matched to patients, because the nucleus transferred could be from one of their own cells. More recently, in November 2006 testimony to the Bioethics Council, Dr. Hans Scholler, Director of Cell and Developmental Biology at the Max Planck Institute in Munster, Germany, discussed further advances that support the scientific feasibility and moral acceptability of ant. Dr. Scholler used ant techniques to preemptively silence a key factor essential for defining an embryonic organism. Using this technique, he was able to obtain pluripotent stem cell lines at the early stages and at a rate 50% higher than from direct destruction of IVF-created embryos. D. Pluripotent stem cells via somatic cell dedifferentiation. Under this proposal, adult stem cells would be reprogrammed to dedifferentiate back into pluripotent stem cells. Crucial to this approach is discovering a way to reverse cell differentiation all the way to pluripotency, but without creating an organism capable of developing nascent life. The preliminary evaluation from the Council in 2005 was that this approach was ethically acceptable for use in humans if and when it became scientifically practical, provided the line between pluripotent cell and living organism can be maintained. The Council noted that while this approach was scientifically and technically uncertain, there were encouraging results appearing in the scientific literature. In 2005, scientists from the Harvard Stem Cell Institute published a peer-reviewed study in which a fusion of human embryonic stem cells with adult stem cells suggested that adult cells could be reprogrammed if the remaining technical barriers are overcome, and some of them are significant. The approaches discussed in this study could circumvent societal concerns over cloning or the destruction of human embryos. A year later, Drs. Takahashi and Yamanaka of Japan published their peer-reviewed mouse study, examining 24 genes as candidates for conveying pluripotency. They discovered that by altering just 4 genetic factors proved sufficient to change adult stem cells into pluripotent stem cells. Scientists will now try to determine if human adult cells can be similarly reprogrammed using this method. In November 2006, scientists from the School of Biosciences and Institute of Genetics at the University of Nottingham reviewed the recent scientific advances in reprogramming and concluded there is sufficient evidence demonstrating that adult cells can be reprogrammed and that cell-based approaches for therapeutics may be a realistic expectation in the future. The study reviewed 9 reprogramming approaches that are commonly used today and commented on their respective advantages and limitations. While it is too early to tell which approaches may or may not work, the sheer number of methods makes clear that there may be several different ways to induce reprogramming in adult cells in the future. Conclusion. As this report shows, there have been tremendous scientific progress of late in exploring methods of deriving pluripotent stem cells without destroying embryos. This groundbreaking alternative research has developed under President Bush's insistence on advancing stem cell research with clear ethical guidelines. With continued support for non-destructive alternatives, new developments will continue to unfold in this field in the years to come, holding the potential for innovative progress towards new medical cures, while at the same time upholding human dignity and the sanctity of innocent life.